Welcome back, everyone. So, in the previous video, we talked about the general process of protein synthesis and all the required structures for it. In today's video, we will be talking about cell transcription, its different enzymes, and the proteins that it requires. Then we will discuss the transcription process in detail: initiation, elongation, and termination. So let us start from where we left off. This was the DNA sequence that we were talking about in the genetic code. Let us make a chain of it. Now in this DNA template, there will be some nucleotide bases in between. This area of nucleotide bases allows us the process of transcription. So let us name them as promoter regions, as in they are promoting the process of transcription. Now remember that transcription occurs in two types of cells: prokaryotic cells and eukaryotic cells. Let us first discuss the prokaryotic cells. In prokaryotic cells, the promoter region binds to RNA polymerase holoenzyme. RNA polymerase holoenzyme has further two parts: core enzyme unit and sigma subunit. Core enzyme subunit has further two alpha chains, two beta chains and one omega chain. Core enzyme then binds with sigma subunit. Sigma subunit will help in binding with the promoter region to initiate transcription. Now this RNA polymerase holoenzyme will be responsible for making RNA inside the prokaryotic nucleus. The enzyme will help to make all three types of RNA, which are ribosomal RNA, messenger RNA, and transfer RNA. But there is a different case with eukaryotic cells. Let us take three promoters here and study them separately. Now in eukaryotic cells, we require two things: RNA polymerase and transcription factors. Here are three promoters, so this means all three RNA will be made separately. Ribosomal RNA will be made separately. Then messenger RNA. And lastly transfer RNA. So on the first promoter region, RNA polymerase will be attached. Let us call this RNA polymerase 1. Then with RNA polymerase 1, there will be an attachment of transcription factor, and this will give rise to ribosomal RNA. Then let's move towards the next promoter. Here, RNA polymerase will bind with transcription factors. Let us name it RNA polymerase 2. Now this will give rise to the formation of messenger RNA. If we want to be more specific, then we can say that RNA polymerase 2 also forms small nuclear RNA as well. This small nuclear RNA is used in the process of splicing. Now let us move toward the third promoter. When it binds to our third RNA polymerase, which we also name as RNA polymerase 3, then it will bind again with the transcription factors and will make transfer RNA. Get a bit more specific. and say that RNA polymerase III also gives rise to small nuclear RNA, which also gives rise to a process of splicing. To remember all of this, have a mnemonic in your mind. One roll to my 3 turtles. 1 means RNA polymerase I. Roll means ribosomal RNA. To mean RNA polymerase II. My means messenger RNA. 3 means RNA polymerase III. And turtle means transfer RNA. If we get more specific, then also remember that RNA polymerase II and III also give rise to the formation of small nuclear RNA. So, this was the intro of the process of transcription. You need to understand the regulation of the process as well. There are 2 types of proteins that increase or decrease the process of transcription. These are enhancers, that will increase the process of transcription, and silencers, which will decrease the process of transcription. If we look at the general structure, here is the promoter region, and here is the enhancer region. Now, what will happen? This enhancer will bend the structure of DNA. There will be an attachment of RNA polymerase and specific transcription factors that will enhance the process of transcription. Same will happen in case of silencers. Here is the promoter region. And here is the silencer region. Can anyone guess what will be the result? This silencer will bend the structure of DNA like this. Same as the process of increasing transcription. There will be an attachment of RNA polymerase and specific transcription factors that will silence or reduce the process of transcription. If you want to understand this better. Now, to understand this process, take an example. You remember thyroid stimulating hormone, right? Well, if we look at its general synthesis as a basic physiology concept, we have seen that TSH of thyroid stimulating hormone give rise to the formation of G protein coupled receptors. We name them GPCR. Then it forms cyclic AMP, which we write as CAMP. Then it gives rise to protein kinase A or pKa. Now this pKa binds with enhancers or silencers. And thus, they either stimulate or reduce the production of thyroid hormone. So, there are 3 steps in transcription. Initiation, elongation, and termination. Let us discuss all of them in the next sections.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.